Welcome to MMBS111, Introductory Microbiology. This is mini lecture 13.6. We're going to be discussing specific immunity and types of immunity and the process of immunization. There are 4 learning outcomes for this mini lecture. First, name the 4 different immune states. Second, explain the characteristics that make for an effective vaccine. Third, name the 2 major categories of vaccines. And finally, what is herd immunity and why is it so important? So there are a number of different immune states depending on how the immunity was acquired. So there is the natural immunity state, and this form of immunity was acquired through normal biological experiences. So typically natural immunity is immunity that occurs because of an active infection. Then we have artificial immunity, which is protection from infection that was obtained through a medical procedure. So we would say that vaccination and immunization falls into this category. It's artificial. Active immunity is a condition where an individual receives an immune stimulus that activates B and T cells. So you have an antigen present that has activated these cells. This also involves the creation of memory. So you're going to have B and T cells that are stimulated that have differentiated into memory cells, so that when you're exposed to that antigen again, there will be a quick response. Active immunity requires several days to develop, and it lasts for a relatively long time depending on the nature of the antigen, and it can be stimulated by natural or artificial means. So active immunity can be stimulated through an active infection, but it can also be stimulated through artificial methods like vaccination. Passive immunity involves the receiving of antibodies from another human or an animal. The recipient is usually protected for a short period of time, even though they've never been exposed to the actual antigen. Passive immunity does not produce memory, so there will not be memory B and T cells produced from this type of immunity, which is why it is temporary. It also does not produce antibodies against the disease, but it does result in immediate protection from that infection. It can be acquired naturally, and passive natural immunity is only acquired between mom and fetus or newborn. It can be acquired artificially. If you've ever donated plasma, that plasma is then used and given to recipients who are not able to produce antibodies themselves. So they can receive those plasma cells from a donor that will then provide some short-term protection from an infection. This image is from your text, and it shows the combination of natural and artificial immunity, along with examples that illustrate active forms and passive forms. So the basic principle behind vaccination is to stimulate a primary response that will then result in production of memory cells. Vaccination is a method that sort of prepares the immune system for future exposure to a pathogen. And this way, if the person is ever exposed to that pathogen, then the response will be much more rapid than if that were the first exposure. So the goal of vaccination is to produce memory cells that will respond immediately the next time that antigen is encountered. There are a number of characteristics that we're looking for in an effective vaccine. So we want a vaccine that's going to protect against exposure to natural wild forms of the pathogen. We also want a vaccine that's going to be safe with very few adverse effects. It needs to be able to stimulate both antibody and cell-mediated responses so that we get memory produced from both cell types. We want it to have long-term lasting effects, so it's important that memory is produced from this vaccine. We want a vaccine that is not going to require numerous doses or a bunch of boosters throughout the course of a person's life. And we also want it to be inexpensive, have a relatively long shelf life, and be easy to administer. There are two types of vaccines that are produced, and each of those has some subtypes. So the first type of vaccine is called a whole cell vaccine, and there are two forms of whole cell vaccines. They are killed cell or inactivated virus whole cell vaccines, or live attenuated cells or viruses. Killed cells or inactivated viruses are not able to produce disease. They are dead or inactive, but the antigens are still present, so they're going to stimulate an immune response. The pathogen is not able to multiply or reproduce, but they are able to stimulate a response from the lymphocytes. A live attenuated cell or virus vaccine is a vaccine that contains live cells or active viruses that have been attenuated. And this means that their virulence has been eliminated or reduced. So they're not going to be able to produce severe disease or infection. They are alive or active, so they're able to reproduce and multiply, which is really an advantage because our immune cells are going to respond as they would if the cell or the virus were in its wild form and still possessed those virulence factors that are going to cause severe disease. So we're going to get longer lasting protection. Live attenuated vaccines are also going to be better at inducing a cell-mediated response from T cells. And they typically last longer and require fewer boosters because we get such better response from the immune cells when the vaccine is a live vaccine instead of killed or inactivated. The other type of vaccine is called a subunit vaccine or a conjugated vaccine. Conjugated vaccines use parts of the pathogen. So it might use certain receptors on the viral capsid, or it might use toxins or toxoids produced by the bacteria, or certain components from the flagella or the cell wall of the bacteria, and then they're conjugated to a larger protein in order to make them more immunogenic. So we want a good response from the immune system. These parts of the pathogen are still antigenic, so or immunogenic, so we're still going to get our immune response, but we're not going to get the production of even mild forms of the disease. So no organism is present, just parts of that organism. So the vaccines have been very successful since their discovery and implementation, and we've actually seen a pretty significant rise in outbreaks of diseases that can be prevented through vaccination, because many people have chosen not to vaccinate themselves or their children. In Idaho in particular, we have a serious whooping cough issue because many people have just chosen not to vaccinate their children. In fact, this year in Canyon County, there were a number of schools that had to report whooping cough incidents. And all of these diseases can be prevented through vaccination. When you choose not to vaccinate, there's a decrease in the level of herd immunity, and part of the goal in vaccinating is to obtain a certain percentage of the population as protected, as vaccinated. This makes it more difficult for the microbe to circulate in the population and be transmitted. It also protects those who cannot be vaccinated, either because of age or health. Individuals who are immune compromised, individuals with autoimmune diseases, the very young and the very old have weakened immune systems or underdeveloped immune systems, and vaccination in the healthy helps to protect those in the population who cannot. It is good for the individual as well as the whole population. In the decade before the measles vaccine was available, there were about 3 to 4 million cases of measles every year. 300 to 400 children died annually, and 1,000 more individuals were chronically disabled by measles encephalitis. According to UNICEF, childhood vaccines save the lives of 2.5 million children a year, and that's worldwide. So there's been a lot of controversy and discussion about vaccinations and if parents should or should not vaccinate, and how do you make that decision. So this debate and controversy really began in 1998 in the UK when a physician, Andrew Wakefield, published a paper in the Lancet that showed a correlation between autism and autism spectrum disorders and the MMR vaccine. That paper was later retracted because other scientists and physicians were unable to replicate Dr. Wayfield's results. And after further investigation, it was determined that his data was faked. So he lost his medical license. However, that one paper has done quite a lot of damage to overall vaccination rates in the UK and in the United States. I've also heard from other individuals that vaccines contain heavy metals and chemicals that are harmful. So I don't want to expose myself or my children to those chemicals, and I'm just not going to vaccinate because they aren't safe. According to the CDC and the World Health Organization, the only chemical that continues to be used in very few forms is thimerosal. It's also known as ethyl mercury. It was removed from all childhood vaccines in 2001. Influenza vaccines are the only vaccines that contain this chemical today, and you can also find this vaccine in thimerosal-free versions. Thimerosal is a preservative, and when you have a vaccine that has multiple doses, thimerosal is used to protect the vaccine from microorganisms that could be exposed to the vaccine during when you're drawing multiple doses from the same vial. While thimerosal is a mercury-containing compound, it is also easily and quickly removed by the body. So there is a lot of debate and discussion, and what I would encourage all individuals to do is to do your research, and especially consider your sources of information. Sources that come from reputable organizations like medical journals, government agencies, universities. Those are relatively trusted sources, and I would look there and do my research and then make my decision based on that, and not on whatever alternative medicine or whatever other parties say. You need to do your own research and make those decisions on your own. Here are some of our checking in questions. So be sure to go through these, make sure that you can answer them correctly. I would encourage you to go back to the beginning of the presentation and review the learning outcomes. Make sure that you feel good about those. Our critical thinking question is here. Using the graph from section 13.5, explain where a vaccination would be most effective and why. So use this graph in your understanding of vaccinations and antibody production. Where would vaccination be most effective? And then explain why you chose that particular time. And formulate your answer. You can come back here and compare your answer to mine on the next slide. And hopefully this video was helpful.